Thanks, Linda, for the kind introduction. I'm also glad to be back. And thank you to the organizers for the opportunity to present the work of the Duke University Consortium for HIV/AIDS Vaccine Discovery, or CHOV-D program that is led by Bart Haynes. So I've divided my talk into two parts. Part one is an overview of HIV-1 vaccine development, where I'll briefly summarize the immunization strategies for eliciting broadly neutralizing antibodies that are referred to as bNabs for short. For part two, I'll present the results of the HVTN-133 human clinical trial that demonstrated vaccine induction of HIV-1 neutralizing B-cell clonal lineages in humans, and most of my talk will be on this trial, or the results of this trial. So as a reminder, a key goal of an HIV-1 vaccine is the induction of broadly neutralizing antibodies, or bNabs, that target the envelope surface protein and prevents infection of multiple different HIV-1 strains. This is a representative structure of an HIV-1 envelope trimer that sits on the surface of the virion and has multiple sites that are targeted by broadly neutralizing antibodies, or bNabs. As a reminder, the trimer is comprised of 3 GP120 and GP41 heterodimers. The blue surface of this image represents the GP120 portion of the trimer, and the beige surface near the base of the trimer represents GP41. Now, there's a portion of GP41 that's membrane-bound and for which we do not yet have a structure, and it's shown at the base of the trimer in cartoons. So as I mentioned, there are multiple sites on this trimer that are targeted by bNabs, and they're shown here in different colors. For example, in purple, connecting GP120 and GP41 is a GP120-GP41 interface. Orange is showing you CD4 binding site. At the top in red is V1, V2 glycan site. And green on the right is V3 glycan, and the dark red in the GP41 portion of the trimer is representing the membrane proximal external region, or EMPER for short. Now, these sites are generally conserved on envelopes from different HIV-1 strains, so antibodies targeting these sites are able to bind to HIV envelopes from multiple different strains and ultimately prevent infection in a process referred to as neutralization that I'll mention throughout my talk. Now, bNabs are also often isolated from chronic human HIV-1 infection, but in the second part of my talk, I'll show you an example of a candidate vaccine-induced bNab B-cell clonal lineage. So Bart Haynes will have a satellite presentation on Tuesday. Bart Haynes will present different strategies for induction of bNabs. The satellite session is titled From Prevention to Cure. This is a schematic showing one of these strategies that is referred to as the prime boosting strategy. So in this strategy, the priming immunogen will stimulate precursor B cells that are capable of developing bNabs over time. However, this bNab development will be dependent on the subsequent boosts, as you need an appropriate boost to expand these B cell lineages and also select for affinity mature B cells that can develop into bNab status. This strategy is widely used in multiple different vaccine clinical trials, and Bart will discuss some of these trials in his satellite session tomorrow. This is a summary of the Duke CHOV-D HIV vaccine clinical trials portfolio from 2023 to 2026. As you can see, there are multiple different immunogens that are being tested in different vaccine strategies as protein ferritin nanoparticle, protein trimer, mRNA, and EMPER peptide liposomes. These immunogens were designed to elicit bNabs targeting different sites, for example, CD4 binding site, V3 glycan, V2 glycan, and EMPER. Bart will describe some of these strategies in the different clinical trials in his session tomorrow, but for the rest of my talk, I'll focus on HVTN-133 that studied repeated immunization of EMPER peptide liposome. So on behalf of the Duke CHOV-D and HVTN-133 teams, I will present the results from HVTN-133 human clinical trial that demonstrated vaccine induction of HIV-1 neutralizing B-cell clonal lineages in humans. I showed you this slide before of a representative structure of an envelope trimer, and here I want to remind you that the membrane proximal external region, or EMPER, is in the GP41 portion of the trimer, and the antibodies that I'll describe in the rest of my talk target this region. So this is a cartoon that summarizes the EMPER broad neutralizing antibody site of action. On the left, in step 1 in antibody binding, you see GP41 EMPER bNab binding to the virus lipid bilayer. And on the right, you see HIV GP41 mediated fusion process of viral entry into CD4 positive cells. GP41 is shown in red, connecting the virus at the top to the cell at the bottom. And during this fusion-dependent process, GP120, shown in blue, gets shed, EMPER on GP41 also becomes exposed. Thus, in step 2 in antibody binding, you'll see that the GP41 EMPER bNab binding to virus lipid bilayer on the left can now bind to the EMPER epitope shown in white as LDKW once EMPER becomes exposed during this viral fusion-mediated process. Once the antibody binds to EMPER, then this ultimately prevents virion infection of these CD4 positive cells in the process referred to as neutralization, as I mentioned earlier. In terms of the properties of bNabs against the conserved EMPER and GP41, previous studies have shown that bNabs that target GP41 EMPER are among the most potent and broad and protect animal models of HIV-1 infection in studies with Shiv-challenged macaques. bNabs that target GP41 EMPER have an epitope that includes membrane lipids, as I alluded to in the previous slide, and EMPER bNabs have hydrophobic HCDR3 loops that are required for lipid binding and neutralization, and I'll get back to this point at the end of my talk. So this is a schematic for the GP41 EMPER peptide liposomes that was studied in HVTN 133. This immunogen is a lipid-based compound that has EMPER peptides attached. And the EMPER peptides contain epitopes that are targeted by the proximal EMPER bNab such as 2F5, distal EMPER bNab such as 4E10, and T helper epitopes. However, for my talk, I'll describe the B-cell responses to this immunogen that was designed by Munir Alam and Bart Ains at Duke University. So in a previous study by Bart and colleagues, they found that the GP41 EMPER peptide liposome elicited 2F5-like precursors in monkeys, and these antibodies targeted the DKW motif of proximal EMPER like 2F5. Shown in the top right of the slide is a structure of one of these antibodies that was isolated from the monkeys, and this antibody was referred to as DH570. In terms of the functional characteristics of DH570, this antibody did not bind lipids and neutralized only in the TZMBL Fc gamma R1 infection assay that was designed to identify EMPER bNab precursors. Now, in the TZMBL Fc gamma R1 assay, David Montefiore and colleagues at Duke engineered TZMBL cells to express Fc gamma R1 or CD64, as shown here in yellow on the TZMBL cells. This allows EMPER antibodies, shown here in red, to use its Fc domain to bind the Fc gamma R1 in the TZMBL cells, and then uses its Fab domain to tether to the viral membrane during the prefusion state of viral entry. As shown on the right, during the pre-hairpin intermediate stage of viral entry, EMPER becomes exposed, and as you can see, an EMPER antibody being tethered to the viral membrane has an increased chance of binding to EMPER upon EMPER exposure. So this assay is particularly important for EMPER bNab precursors because they have not yet fully affinity matured, so they cannot bind lipids in the viral membrane on its own and await the exposure of EMPER during viral entry, unlike the mature EMPER bNabs. So this is a summary of the degree of proximal EMPER bNab lineage maturation induced by GP41 EMPER peptide liposomes in monkeys. On the top, going from left to right, are antibodies at different stages of the EMPER bNab lineage maturation pathway, and the characteristics are listed below each antibody. For example, on the left, you see the unmutated ancestor or near germline antibody. This antibody does not bind lipids, but it binds the DKW epitope. And all the antibodies at the different stages of the EMPER bNab lineage maturation pathway binds the DKW epitope. The early intermediate is like the unmutated ancestor, but it can neutralize, easy to neutralize, or tier 2 HIV strains in the TZMBL Fc gamma R1 assay that I described in the previous slide. And the tier 1 viruses are those that often have envelopes with an open conformation, so they're more neutralization sensitive. The late intermediate has weak lipid binding. This allows the antibody to bind to lipids in the viral membrane and await EMPER exposure during the fusion process of viral entry. But this weak lipid binding is inefficient or not as efficient as bNabs. However, the late intermediates can also neutralize the easy to neutralize tier 1 HIV strains in the standard TZMBL assay. And finally, the bNabs have strong lipid binding, so they can bind to the lipids in the viral membrane and await EMPER during viral-mediated fusion process. And these bNabs are associated with neutralization of the difficult-to-neutralize tier 2 HIV strains in the standard TZMBL assay. And the tier 2 viruses often have envelopes with a closed conformation. So neutralization of these viruses is an assessment of neutralization breadth of bNabs. We hypothesized that the DH570 antibodies that were elicited by GP41 emperor peptide liposomes in monkeys were early intermediates in the emperor bNab lineage maturation pathway. And at the end of my talk, I'll show you at which stage of this maturation pathway were the antibodies that were elicited by GP41 emperor peptide liposomes in humans. So this is the immunization schedule for HVTN133 human clinical trial that studied GP41 emperor peptide liposomes. The vaccine trial participants were scheduled to receive 4 immunizations spanning 12 months. However, the trial was halted after an apparent PEG anaphylaxis reaction in one of the vaccine recipients within hours after the 3rd immunization. As a result, all the vaccine trial participants received the first 2 immunizations, but only a small subset of the vaccine recipients received the 3rd immunization. In terms of the immunogenicity of GP41 emperor peptide liposome in HVTN133 after 2 immunizations, Georgia Tamaris' group at Duke found that 95% of the vaccine recipients had serum responses to vaccine-matched emperor peptide. Bart Haynes' group at Duke showed that serum antibodies targeted nominal DKW proximal emperor bNab epitope, also targeted by 2F5, in 65% of the vaccine recipients. And Julie McElrath's group at the Fred Hutch found that emperor bNab phenotype memory B cells were present in 35% of the vaccine recipients. So these were CD19 positive, IgG negative, CD27 all memory B cells that bound to a wild-type emperor peptide, but not a mutant peptide with a disruption of the emperor bNab epitopes. And these memory B cells were found at a frequency of 0.03% of the emperor positive IgG memory B cells. Now, given these outcomes, we next sought to characterize the B cell repertoires in humans following immunization with HIV-1 GP41 emperor peptide liposome immunogen. For our methods, we isolated antigen-specific or emperor-positive B cells from blood of vaccinees post-3rd immunization, and I'll describe the strategy in more detail in the next slide. Following the antigen-specific sort, we expressed recombinant monoclonal antibodies, or MOABs, that were derived from the sorted emperor-positive B cells. We then tested these antibodies for binding specificities, functions, and structure. So this flowchart summarizes the high-throughput strategy for isolation and production of recombinant antibodies. So first, we start with a flow cytometry-based approach to isolate these antigen-specific B cells. A single cell gets sorted per well of a 96-well plate. We then use PCR-based approaches to amplify the antibody heavy and light chain genes. By sequencing these genes, we get immunogenetics information that can be used to make inferences about antibody function. We also use these heavy and light chain gene amplicons from each B cell and clone them into a linear cassette using overlapping PCR. Using these linear cassettes, we can transfect cells, 293 cells in 6-well plates, so we generate microgram quantities of antibodies for a pre-screen assay, which is generally a binding assay, and we select the best binders and express them in bulk in milligram quantities for further functional characterization. This is a representative flow cytometry sort plot demonstrating candidate emperor-positive B cells isolated after 3 immunizations in one representative HVTN133 vaccine recipient. The blue dots shown in the gate in the upper right-hand quadrant of the plot represents the candidate emperor-positive B cells that bound emperor peptides conjugated to 2 different fluorophores for flow cytometry sorting. In total, we sorted 510 CD19 positive B cells from 5 vaccinees after 3 immunizations. We recovered 87 high-affinity emperor-positive antibodies and expressed 80 of them as recombinant monoclonal antibodies for functional characterizations. These 80 antibodies represented 38 unique B cell clones, where a single clone contains B cells that were computationally inferred to be derived from the same parent B cell. Among the 38 B cell clones, 24 clones had B cells that generated antibodies that can neutralize HIV in the TZMBL Fc gamma R1 assay that I described earlier. 14 of 38 clones had antibodies that neutralize the easy to neutralize tier 1 HIV in the standard TZMBL assay. And strikingly, 2 of 38 clones had antibodies that can neutralize the difficult to neutralize tier 2 HIV strains in the standard TZMBL assay, which is characteristic of bNabs. And later on in my talk, I'll describe one of these bNab lineages in more detail, and this bNab lineage was referred to as DH1317. Now, among the sorted B cells after 3 immunizations were emperor bNab phenotype antibodies. They bound to a wild-type emperor peptide, but not a mutant peptide with a disruption of the emperor bNab epitope. These emperor bNab phenotype antibodies were found in 2 of our best responders at a frequency of approximately 1 per 15,000 total B cells post-3rd immunization, in contrast to 0 per 1 million total B cells. This is a list of the predominant heavy chain genes that were used by the EMPER-positive antibodies in vaccine recipients post-third immunization. And as you can see, VH7-4-1 was the most frequently used by 32% of the EMPER-positive antibodies after three immunizations. In contrast, we found that these heavy chain genes were used at substantially lower frequencies in the antigen unbiased B-cell receptor repertoire at baseline in these 5 vaccine recipients that we studied. Therefore, these data suggested that the EMPER-positive antibodies that use these heavy chain genes after 3 immunizations were preferentially selected by the vaccine. This is a list of the heavy chain genes used by antibodies in the 14 EMPER-positive B-cell clones that neutralized HIV in the standard TZMBL assay, which is indicative of a polyclonal heterologous neutralizing antibody response. There were 7 clones that used VH7-4-1 with HCDR3s ranging from 14 to 23 amino acids. 5 clones used VH5-51. HCDR3s ranged from 13 to 26 amino acids. There was 1 clone that used VH1-69 with 18 amino acids long HCDR3, and another clone that used VH3-49 with 9 amino acids long HCDR3. So here we show that their neutralizing EMPER-positive B-cell clones use varied HCDR3 lengths, some of which were also used by EMPER-positive bNabs that were previously isolated from chronic human HIV-1 infection. So this is the phylogram of a heterologous HIV-1 neutralizing VH7-4-1 using B-cell clonal lineage that we referred to as DH1317. This is an 11-member IgG clone. All the antibodies use VH7-4-1 with 15 amino acids long HCDR3 and VKappa1-12 light chain. Shown in the black circle is DH1317.4, which is our best neutralizing antibody in this clone. We found that DH1317.4 neutralized 15% of a panel of 197 multiclade heterologous HIV-1 strains that were tested in the standard TZMBL assay by David Montefiore at Duke and Mike Seaman at Harvard. The neutralization titers range from 0.1 to 48 microgram per ml. The smaller the number, the better the neutralization titer. And the geometric mean IC50 neutralization titer was approximately 13 microgram per ml. This antibody also demonstrated a preference for neutralizing clade B HIV-1 strains in that it neutralized 35% of the 72 clade B strains that were present among the 197 viruses tested, and the geometric mean IC50 neutralization titer was also 13 microgram per ml. Additionally, we find that in the standard TZMBL assay, DH1317.4 did not neutralize any of 75 clade C HIV-1 strains, which is similar to the 2F5 prototype proximal EMPER bNab. In contrast, in the TZMBL FC gamma R1 assay, DH1317.4 neutralized clade B Amtrial breakthrough viruses that are difficult to neutralize, as well as clade C and other non-clade B HIV-1 strains, demonstrating that the paratope for DH1317 antibody recognition of EMPER on multiclade HIV-1 strains was intact. So next, I'd like to demonstrate the progression of neutralization capacity in HVTN133 vaccine-induced heterologous HIV-1 neutralizing B-cell clonal lineage. I showed you this phylogram earlier of the DH1317 lineage, and here I want to point out that as shown by the black box, the UCA or near germline antibody neutralizes HIV only in the TZMBL FC gamma R1 assay, and thus is an early intermediate in the EMPER bNab lineage maturation pathway. The green boxes show the antibodies that can also neutralize the easy-to-neutralize tier 1 HIV in the standard TZMBL assay, and these are indicative of late intermediates in the EMPER bNab lineage maturation pathway. And finally, the red boxes show the antibodies that can also neutralize the difficult-to-neutralize or tier 2 HIV-1 strains in the standard TZMBL assay, and these are indicative of early bNabs in the EMPER bNab lineage maturation pathway. So here I've demonstrated the progression from precursors to the development of heterologous HIV-1 neutralization activity, which is characteristic of bNabs within a single vaccine-induced B-cell clonal lineage, which has never been reported. In terms of the structural analysis of DH1317.4 bNab, this is work done by the structural biology team at the Duke Human Vaccine Institute. On the left is a cryo-EM 3D reconstruction of a HIV-1 envelope trimer, where the conformation mimics that of the envelope in the pre-fusion state. This trimer was also engineered to express EMPER as shown at the bottom. And on the right, you're seeing a negative stain electron microscopy map of DH1317.4 bNab in complex with this trimer. The side view of this complex is shown at the top, the bottom view at the bottom. You see this trimer in gray, the EMPER portion of the trimer is in green, and in light red is the DH1317.4 Fab binding to EMPER on the SOSIP trimer. So previously, or earlier in the talk, I mentioned that previous studies have shown that Emperor bNabs have hydrophobic HCDR3 loops that are required for lipid binding and neutralization. So we next wondered what was the relationship between lipid reactivity and neutralization of the DH1317 lineage antibodies. So these are biolayer interferometry or BLI data from Munir Alam at Duke showing DH1317 lineage antibodies binding to liposomes containing binary lipids on the x-axis, or the different DH1317 mature antibody, as well as the unmutated common ancestor or near germline antibody on the far left. And on the far right is our positive control, which is the prototype proximal Emperor bNab 2A5. There's normalized response, or in this case, lipid binding on the y-axis, where the higher the number, the better the binding. And as shown by the red asterisks, you'll see that the best neutralizing antibodies in this clone demonstrated the highest lipid binding. This graph shows a correlation between the lipid binding and neutralization of DH1317 antibodies. So there's lipid binding levels on the y-axis. The higher the number, the better the binding. On the x-axis, this is neutralization of a representative heterologous HIV-1 strain, where the smaller the number, the better the neutralization titer. Each circle represents a mature DH1317 antibody, the UCA shown on the far right. And here you can see a nice correlation between lipid binding and neutralization of the DH1317 clonal lineage antibodies. And finally, our computational biology team at DHVI generated lipid insertion propensity scores for the DH1317 lineage antibodies. So here they calculated what is referred to as a delta G whiff score. So as shown on the y-axis, you'll see the delta G whiff score, which is a measure of the hydrophobicity of the HCDRs within the antibody. And the lower the delta G whiff score, that means the HCDRs are more hydrophobic, and so they have an increased lipid insertion propensity. On the x-axis, you're looking at neutralization titer of a representative HIV-1 strain, and again, the smaller the number, the better the neutralization titer. So as you can see, you have the antibodies with the smaller number in terms of IC50, the better neutralization capacity, also have lower delta G whiff scores, demonstrating that they have more likely hydrophobic HCDRs, so increased propensity for lipid insertion, and they're more likely to bind the lipids in the viral membrane and await Emperor exposure during viral entry to block these viruses via neutralization. So I showed you this slide earlier, and here I want to highlight that the antibodies that were elicited by GP41 Emperor peptide liposomes in humans demonstrated characteristics that were more like late intermediates or early bNabs. For example, DH1317 lineage antibodies that demonstrated neutralization of difficult to neutralize tier 2 HIV in the standard TZMBL assay. So in summary, GP41 Emperor peptide liposomes induced a polyclonal neutralizing antibody response ranging from early and late intermediate antibodies to early bNabs in HVTN133. We isolated a large bNab clonal lineage termed DH1317 that demonstrated the full range of neutralization development from precursors to heterologous HIV-1 neutralization status. Structural analyses of DH1317 antibodies revealed binding to Emperor in the context of HIV-1 envelope trimer. DH1317 antibodies also bound a similar epitope to that seen with GP41 Emperor peptide liposome-induced bNab precursors in monkeys, and I didn't have time to show you those data. However, I showed you that DH1317 neutralizing antibodies demonstrated 15% neutralization breadth against a panel of 197 multi-clade heterologous HIV-1 strains, with preference for clade B strains at 35% breadth. And we hypothesized that the lack of additional breadth is due to insufficient bNab affinity maturation and insufficient recognition of HIV sequence diversity. Finally, lipid binding of DH1317 clonal lineage antibodies correlated with neutralization potency, as I showed you. However, some DH1317 antibodies had limited polyreactivity, and I didn't have time to show you those data. So in conclusion, we've demonstrated that proximal Emperor bNab precursors can be activated by vaccination in humans to neutralize HIV, that a single immunogen could induce Emperor epitope DKW targeted antibodies to neutralize, suggests that repeated immunization with the same immunogen could select for affinity matured antibodies with greater bNab breadth and potency. And finally, future studies will reformulate GP41 Emperor peptide liposomes without PEG and design immunogens that target both proximal and distal Emperor bNab epitopes. I would like to end by acknowledging different members of the Duke Human Vaccine Institute that's led by Bart Ains at Duke University, members of my lab that was involved in this project, different labs at Duke, as well as our collaborators at other institutions, the HVTN 133 trial participants, and our funders to whom we're most grateful. Thank you very much for your attention.